My name is Dr. Christian Lambert and I'm a consultant neurologist and clinician scientist based at the Wellcome Centre for Human Neuroimaging. One of the problems we've got in trying to study behaviour and disease in the brain is the huge amount of variability observed between individuals. In trying to account for this variability, the possible causes can span many different biological scales. The work that I do tries to anchor this in studying brain anatomy, because cortical structures provide relatively stable features that we can readily measure and quantify in vivo, and certainly in some cases provide us with causal associations. One of the problems we've got in measuring individual level anatomy in vivo is that the current methods available to us significantly underestimate the true amount of individual variance. This project aims to build upon a body of methods we've been developing to map these anatomical structures in individuals. The aim is to then apply these to neurodegenerative conditions such as Parkinson's disease to enable us to better predict individual level patient trajectories and also diagnose the disease at a much earlier stage. I'd now like to hand over to Professor John Ashburner, who's going to explain the engineering aspects of this project.
Hello, I'm John Ashburner and I'll be leading the engineering aspects of the project. So the idea will be to begin with the MRES part of the project, which will be to improve the quality of the quantitative maps that we'll be using during the rest of the project. The next part of the project will involve combining the high-resolution quantitative images that we acquire with slightly lower resolution diffusion data to try and do a clustering approach that combines the information from both the diffusion images and the quantitative maps and achieves results in a relatively high resolution. So the segmentations of the high quality images that are achieved in the second part of the project, along with simulated images that can be generated using the quantitative maps, they can both be used for training deep learning algorithms to segment a wide variety of different types of MR scans. So this kind of approach could be applied to very large data sets such as that acquired by the UK Biobank. The ultimate aim of the project, however, is to be able to make predictions about individuals. So if we can predict, based on their brain anatomy, who will, say, convert to Parkinson's disease or not, then that would be a useful clinical addition to the work.
I'm Mikkel Brutt and I am a third-year PhD student working here mainly with Professor John Ashburner as my technical supervisor. I think working with John and Chris is also a good opportunity because they have great knowledge. They both have strong technical knowledge, obviously, and Chris, as well being a consultant neurologist, knows a lot about the clinical aspects. I decided to do my PhD at UCL because, well, first, it is a great university. Second, it's a very international university as well. You meet people from everywhere. Third, there are immense opportunities for collaborations, and almost daily there are interesting talks, journal clubs to attend, and London is an exciting city to live in.